welcome to my lesson on the PAG4 experiment investigating factors affecting the rate of photosynthesis. For this investigation, we will use the leaf disc experiment. The aim of the experiment is to use a quantitative test to investigate how changing light intensity affects the rate of photosynthesis. The independent variable in this experiment will be the light intensity, which will be changed by changing the distance between the lamp and the apparatus, and the light intensity will be measured in lux. The dependent variable is how long it takes for all of the leaf discs to rise to the top of the syringe. This is measured using a stopwatch and recorded in seconds. Finally, the graph will be drawn of lux versus rate of movement in millimetres per second. This experiment can be done at home, as you should be able to get hold of all of these apparatus. Since oxygen is the waste gas of photosynthesis, the rate of release of oxygen is comparative or represents the rate of photosynthesis. Using the leaf discs, if you draw the air out of the air spaces between the spongy mesophyll cells by using a vacuum, by putting your thumb over the top of the syringe and pulling repeatedly down on the plunger, then you can replace the gases with water, and this makes the leaf discs sink. Then, as oxygen is released during photosynthesis, the oxygen goes into the air spaces again and also is released from the stomata and get trapped underneath the leaf disc. This then makes the leaf discs float. However, obviously aerobic respiration is also occurring at the same time and using up oxygen. So the two processes counter each other, with the accumulation of oxygen in the air spaces. So the rate of rising of the leaf disc is actually an indirect measurement of the net rate of photosynthesis occurring in the leaf discs. The rate of oxygen release can be compared for different light intensities by timing how long it takes for the leaf discs to float.
For this experiment, we're trying to investigate how light intensity affects the rate of photosynthesis. Now, as we know, carbon dioxide is required for photosynthesis, and we're going to be looking at plant leaf discs. And basically, we're going to look at, to measure the rate at which they photosynthesize, we're going to put leaf discs into a solution of water with bicarbonate of soda in it to provide the carbon dioxide for photosynthesis. So I've got bicarbonate of soda there, and I just need to put 1/4 of a teaspoon into about 300 mils of water. And then that provides the carbon dioxide. And then we need to add just 1 drop of liquid soap, and that helps draw the oxygen out of the leaf.
The baking powder, when added, provides the leaf discs with an alternate source of carbon dioxide in the form of bicarbonate ions, so photosynthesis can occur in the sunken leaf discs. You must be careful when adding the soap that suds do not form. The soap acts as a surfactant for the hydrophobic surface of the leaf, allowing the bicarbonate solution to be drawn into the leaf, and so the leaf disc can sink. This is important as it is quite tricky to get the leaf discs to sink.
So what we're going to do is we're going to see how quickly leaf discs can float to the top of this syringe. So what we've got to do is we've got to make some leaf discs and then draw all the oxygen that is sitting in the air spaces in the leaf out. And if we can draw the oxygen out of those air spaces, it means that they will sink. As the leaf discs photosynthesize, they release oxygen, which gets trapped underneath the leaf, and it makes the leaf rise. And the quicker the leaf rises, the faster the rate of photosynthesis. So I have 10 discs that I have cut out of an ivy leaf using a hole punch. And those little discs are, you just need to take the little discs and you put them into your syringe. Put them in. I've put them in the water from there, so I'll pour them in. Try to get a little bit more out. There we go. So now I've got the plunger, which goes into the syringe. And then I can expel some of the water, and I can get them, and then what I do to get them down to the bottom, I have to suck, by pulling the plunger, I suck the oxygen out, and hopefully that brings the leaf discs down to the bottom of the syringe. And then what we need to do is make sure we get rid of that air, get rid of the air, and then draw in enough water to make 20 mils. And what we're going to do is we're going to put the light source at different distances from the syringe. And then we will measure how long it takes for these leaf discs to float to the top. Now, it does take quite a long time, so I will show you how I set it up, and then I will put the results on the video. So what I do is I'm going to be doing 0, 5, 10, 15, and 20. So, because 0, 5, 10, 15, 20 gives us 5 different distances. I will then measure the lux at each of those distances using an app I have on my phone. And then we can have a look at the rate at which the leaves move. And so you can either use 1 over time to plot it, or we can do the distance that they've actually moved. So this is 6.5 centimetres, so they will be moving 6.5 centimetre in a certain time. And so therefore we can work out rates of centimetre per minute or second. And we can plot that against the lux.
So what we need to do is ideally shut the curtains, and then we will measure the distance. It does take some time, so you can put the syringe down at 0 so that it is directly under the light, so the light is shining onto the leaf discs, and then they will start to photosynthesize, produce oxygen, and then they should hopefully end up rising after several minutes. Once all the leaf discs have risen to the top, record the time. Then, before repeating or going on to the next distance, you need to repeat the process of drawing the air out of the leaves so that they sink again. These are my results for the experiment, and we worked out the rate because we knew that the length of the syringe was 65 millimetres, so 65 divided by the time taken for the disc to rise gives you the rate of movement of the discs. As light intensity increases, so does the rate of movement of the discs, as shown by the graph. The rate of the movement of the discs indicate a rate of increase in the photosynthesis, as the discs rise due to oxygen gas being released into the air spaces in the spongy mesophyll layer, and also being released from the stomata and getting trapped under the discs. The increased light intensity increases the light energy being absorbed at photosystem 2 and photosystem 1, where electrons from chlorophyll are being excited. These electrons get collected by electron acceptors and pass down the electron transport chain. As the electrons are excited and leave the reaction centres of the photosystems, they are replaced by electrons from photolysis. Photolysis requires light energy to occur, photo meaning light and lysis to split. Photolysis is the splitting of water. As the water is split, 2 electrons and 2 protons are released, which are used in the light-dependent reaction, and half an oxygen molecule is released as a waste gas. The oxygen is the waste gas of the light-dependent reaction and is released by the cells. The greater the light energy, the greater the number of electrons excited, and the greater the rate of photolysis of water. Therefore, the greater the rate of release of energy.